All right, an introduction to genetic technology. That's better. Clones. Everybody's heard the word clones. If you're a Star Wars fan, you know what clones are. They are genetically identical molecules, cells, or organisms, all derived from a single ancestor. And the process of producing identical copies of these biological entities is called cloning. And a couple different cloning methods, embryo splitting and nuclear transfer. So embryo splitting, an unfertilized egg is collected from a donor mother. So females, especially female mammals, have all the eggs that we will ever have as soon as we're born. And so an egg is collected from a donor mother, then fertilized in a dish by in vitro fertilization. That's the process that infertile couples can go through to have a child. The fertilized egg develops in the dish to form an embryo containing 4 to 8 cells. After the cells are separated from one another, they divide to form genetically identical embryos. So the fertilization happens, and then that cell divides into 2, and then those 2 cells divide into 4, those 4 cells divide into 8. The scientists separate those cells. And all those cells are going to develop into genetically identical embryos. Those embryos are implanted into surrogate mothers for development. And this is a variation on nature's way of producing identical twins or triplets, and can be used to clone any mammalian embryo, including human embryos. It has not been done with human embryos yet, at least that we know of. Maybe in some dark lab somewhere it's been done, but in the light of day, in the light of the scientific community, humans have not been cloned, but this is how they know how they would do it. An enucleated egg, this is the process. Enucleated means the nucleus has been removed. Provides an environment for embryonic growth. The nucleus is removed to eliminate genetic contribution from the egg donor. And so they did this with a sheep, which we'll see in a minute. You can see the egg cell from one sheep. A donor cell is taken from the sheep's udder. And then an egg cell is taken from an adult female sheep, a completely different female sheep. The nucleus of the egg is removed from the adult female sheep, from the black-faced sheep. And then they fuse the donor cell with the surrogate cell. So it contains the nucleus of one sheep, but the egg of another sheep, form an embryo. Embryo is transferred to the uterus of a foster mother, a surrogate mother. The embryo developed into Dolly, which was the first cloned mammal ever. Happened in 1997, I think. So Dolly was the first mammal cloned by fusion of a nucleus from an adult, a differentiated cell with an enucleated egg. This is a little blurb on Dolly.
Dolly the sheep was a sensation when she first appeared before the world. A scientific breakthrough, fascinating, but also menacing. Dolly was cloned by taking a body cell from sheep A, fusing that cell's DNA with an egg cell with its nucleus removed from sheep B. This fused cell developed into an embryo, which was placed in the uterus of a surrogate mother, sheep C. The resulting lamb, in this case Dolly, is a clone of sheep A. These were Dolly's so-called siblings, identical sisters, all cloned from the same sheep. Professor Kevin Sinclair worked with them and knew Dolly.
It was a step change in our understanding of early development, which ultimately led several years later in 2006 to the discovery that you can actually induce somatic cells. These are sort of cells that belong to the body to become embryonic-like cells that has the potential to divide into other cell types.
This was Dolly's greatest legacy, advances in what we call stem cell research. The ultimate, but still elusive goal is to use cloning technology to create healthy tissues which can be transplanted to heal damaged cells and organs. But back in 1997, Dolly seemed to raise more frightening issues. If we can clone a sheep, many asked, why not a human being? Since then, scientists have cloned more than 20 species, including horses and household pets. But scientists have also recoiled from using these technologies on humans.
I think where we draw the line is if we're going to use genetic manipulations, we have to be extremely careful, and we have to look for severe disease effects to control for. Because with a lot of these things, it's not just the person you're doing a genetic manipulation on or potentially cloning, but it would affect all the generations ahead.
So Dolly's brave new world has not quite turned out as predicted. A sheep created like none before her, but she could not escape her own mortality. She died in 2003, now stuffed in a Scottish museum. Barnaby Phillips, Al Jazeera, London.
So that was Dolly the sheep. We actually clone more genes than we do entire organisms. So recombinant DNA technology is a set of techniques used to clone genes. This revolutionized laboratory research, healthcare, and the food we eat. We can locate genes, map them, isolate them, sequence them, and transfer them between species. We could actually do it if we were in a classroom. Recombinant DNA technology is used to identify carriers of genetic disorders, perform gene therapy, and create disease-resistant food plants. Your assignments this week are on the disease-resistant food plants. Is that a good thing? The immediate goal of cloning DNA, including genes, is to produce a large number of identical molecules, all of which are copies of one ancestral molecule. So we've talked a lot about chromosomes and genes in chromosomes having some sort of mutation leading to some sort of disease. Well, scientists' goal is to just go into that one chromosome and fix it or replace it. And this technology is helping them do that. So to clone a gene, you have to cut DNA into fragments. You do that using something called restriction enzymes. Then you take DNA ligase, glue these DNA fragments into a carrier molecule for transfer into host cells. DNA ligase is another enzyme, ends in ASE. The ligase is always like a glue. And then you grow the host cells to obtain large quantities of the cloned DNA for study. So you don't have to, we have 3 million base pairs. Well, if we only want to study 50 base pairs, you got to be able to take it out of that 3 million and study it. So this is how they do it. They just cut a segment out and they amplify it or copy it many, many, many times to be able to study it. Restriction enzymes come from bacteria. There are bacterial enzymes. They cut at specific DNA sequences. Those specific DNA sequences are called recognition sites. Some enzymes produce overhanging single-stranded ends like you see in the picture below. They're called sticky ends. And 2 DNA fragments with complementary sticky ends will combine or hybridize by base pairing. So restriction enzymes are like a little pair of scissors. They'll come through, cut DNA at a specific sequence. Different restriction enzymes have different sequences that they recognize. And they just snip, cut it off. And sometimes it's not even. Sometimes you get these overhanging ends like you see in the picture. Those sticky ends. common restriction enzymes. These are just pictures. Circle some. You have EcoR1 is an enzyme, and it'll come and cut here. So it ends up like that. You get your sticky ends. SMA1 is a different enzyme. This one just cuts straight through. These are called blunt ends. HAE3 is a different restriction enzyme. BAM-HI, that's a different restriction enzyme. So these are just 4 different enzymes and their sites that they cut, the recognition sites that they recognize and cut. Vectors serve as carriers of the DNA to be cloned. These are plasmids. So once a collection of DNA fragments has been produced, the next step is to link those pieces with carrier molecules for transfer to a host cell. Many species of bacteria carry small circular DNA molecules called plasmids. They're separate from their main nucleoid area. During cell division, the bacterial chromosome and plasmids are copied so that each daughter cell receives a chromosome and at least one copy of the cell's plasmids. By genetically engineering plasmids, researchers developed carrier molecules called vectors that can transfer foreign DNA sequences into bacterial host cells for cloning. Types of vectors, like I said, bacterial plasmids. We can also use yeast artificial chromosomes called YACs. We do this for large cloning projects such as the Human Genome Project. But that's just a simplified bacterial cell. You have the regular bacterial DNA, and then you have these tiny little circles of DNA called plasmids. And that's most of the time what we use as vectors, unless it's a very, very large project, then we use yeast. So this is just a GIF that goes over. You have the bacterium, then you have the plasmid. Then you have the cell containing the gene of interest, it's a completely different cell. Cut a strand out. And then the restriction enzyme comes, cuts its DNA sequences, opens the sticky ends up. And then the gene of interest can be inserted into the plasmid. DNA ligase is the glue. So you have the bacterial DNA mixed with something else, and that's called a recombinant DNA plasmid. So that's a GIF. You can watch it over and over. And then you have to grow the bacterial cells containing the recombinant DNA molecules. So to create a recombinant molecule for cloning, DNA from human cells is cut with a restriction enzyme, producing sticky ends. The vector is cut with the same enzyme, also producing sticky ends, but you want those sticky ends to match, so it's important to use the same enzyme. The human DNA fragments and the cut vector are mixed together. In many cases, the human DNA fragments and vector molecules will combine to form recombinant plasmids, like we just saw in that GIF. Contains human DNA and plasmid DNA, all mixed together. Once a DNA fragment and the vector have been linked together by ligase, the resulting recombinant molecule is ready to be cloned. Bacterial cells containing the recombinant DNA molecules are grown in petri dishes. Bacterial colonies that grow derive from a single cell, which means they are all clones. So in class, in my medical interventions class especially, but we could do it in this class too if we were in person, we take a jellyfish gene, it's called GFP, and makes jellyfish glow. Some jellyfish glow underwater in the dark, and we can insert that gene into bacteria, and it makes the bacteria glow. So we can grow glow-in-the-dark bacteria. So it's not that hard. We can do it in the classroom. Cloned libraries. There's genomic libraries, chromosomal libraries, and expressed sequence libraries. And what is a library? It's a place that you go to get information. So a genomic library is a collection of clones representing all of the DNA sequences of an individual. Chromosomal library is a collection of clones representing all of the genes from one chromosome. So for us, we would have 46 different libraries for our 46 chromosomes. An expressed sequence library is a collection of clones representing the expressed genes from a given cell type. Those are brain cells, express different genes than our eye cells, than our stomach cells, than our hair cells. They all express different genes. That's what makes them different. Then we have to go back and find the different genes in the library. And just like a library that has millions of books, you got to have a way to find a specific book. You have to have a way to go back and find a specific gene within that library. So we have 20,000 genes. Library screening is a method of searching a library for a specific gene using a specifically designed probe. A probe is a labeled single-stranded nucleic acid, and it will base pair or hybridize with complementary sequences in a library. So out of those 20,000 genes, we stick a probe in there with a specific sequence to find the sequence that we want. And the probe will usually have a chemical or radioactive label that will reveal the presence of a bound probe in the library. So we can make it glow. And then we can scan the entire genome to see which area glows, and that would be the area that the probe attached to. So this is the process. Library screening part 1, grow bacterial cells containing DNA clones. Individual bacterial cells from a DNA library are spread over the surface of a solid growth medium in a petri dish. The cells divide and form colonies, clusters of millions of genetically identical daughter cells. So that looks like about 100 clusters, but each one of those has 1 million different bacterial cells in it. A DNA binding filter pressed onto the surface of the growth medium will bind some cells from each colony. The filter is soaked in a solution that ruptures the cells and releases their DNA. The The DNA clings to the paper in spots mirroring the distribution of colonies. And a radioactive probe is added to the liquid. The probe hybridizes or sticks to the only spots of DNA that contain the complementary base sequences. So the probe itself is a single-stranded nucleic acid with a certain set of bases. And it wants to find its matches. If it's an A, it wants to find a T. If it's a C, it wants to find a G. It scatters throughout the liquid, finding its partners in the right order. And it's radioactive, so you take an X-ray. So here, one radioactive spot darkens the X-ray film. The position of the spot on the film is compared to the positions of all the original bacterial colonies. Cells from that colony that made the spot are cultured, and the DNA they contain is harvested. So we can tell just from the radioactive probe, that's the spot we want. So we're going to take that spot and harvest that DNA. A PCR, polymerase chain reaction. Now, if you've heard them talk about coronavirus testing, the fastest coronavirus test that you can take is called a PCR test. That's what this is. This is how they test if you have the coronavirus. Amplification of DNA segments rather than an entire DNA molecule. It's a process of multiple cycles of in vitro DNA synthesis. Does not use restriction enzymes, vectors, or host cells. And its amplification is possible from very small amounts of starting material, the DNA. This is the process that crime scene investigators go through when there's only a tiny, tiny little blood at a crime scene or a tiny, tiny little bit of DNA at a crime scene. They can put that DNA through this process to make millions more copies of it. And this process has replaced host cell cloning in many places. Because why clone a whole cell if you can just take the segment you want and copy that? So each PCR doubles the amount of DNA with each cycle. 1 goes to 2, 2 goes to 4, 4 goes to 8, and so on. So after 30 cycles, there's almost a million copies, over a million copies.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So that's the PCR process.
Polymerase chain reaction, or
There we go. And then you have to analyze the clone sequences. You have to know what you're doing. So you can't just copy it and leave it alone. You have to see if you did it right, see if you got the right gene. So it's used for uses for identified clone sequences. Probes. You can make probes out of these cloned sequences to study sequences on adjacent chromosome regions. You can also use them to isolate the same gene from another species. So if we want to figure out if mice have the same gene that we do for skin color, you create a probe for that. You can also use them to investigate gene structure or study gene expression patterns. Methods for isolating and analyzing cloned sequences. One is called the Southern blot technique, isolation of DNA sequences by gel electrophoresis, followed by identification with the probes. Or just simple DNA sequencing determines the nucleotide sequence. This is the Southern blot sequence.
These 4 cells represent different clinical isolates of the same species of bacteria. Each contains plasmid DNA, but the plasmids in one isolate may not be the same as the plasmids in another isolate. We can purify the plasmid DNA from the isolates and use 2 procedures, gel electrophoresis and Southern blotting, to compare these plasmids. As a first step, we can use gel electrophoresis to examine the plasmids in their native form. Blue loading buffer is added to an aliquot of the plasmid sample, and the mixture is loaded into wells of a jello-like slab made of a polysaccharide material called agarose. The agarose contains microscopic pores through which the DNA can travel. An electric current draws the negatively charged DNA through the gel toward the positive electrode. The positions of the DNA on the agarose gel indicate the sizes of the plasmids. Smaller pieces of DNA migrate faster than larger ones through the agarose pores, because larger pieces meet with more resistance. In this example, the plasmids in lanes A through D are similar in size. The plasmid in lane C migrates slower than the others and is therefore a little larger. Although the plasmids in A, B, and D appear to be the same, we must perform other tests before we can come to this conclusion. Our next step is to add enzymes called restriction enzymes to the plasmids. An example of a restriction enzyme is EcoR1. A restriction enzyme cuts the DNA at a particular recognition sequence. EcoR1 recognizes and cuts the sequence GAA-TTC. If the plasmids in the different tubes are identical in sequence, they should have the same number of EcoR1 recognition sequences. The type of plasmid in this particular sample can be cut in 4 places with EcoR1, producing 4 fragments of different sizes, represented here as different colors. When the fragments are loaded on a gel and electrophoresed, the fragments separate from each other. The slower bands on the gel contain larger fragments, and the faster bands contain smaller fragments. The patterns of fragments in lanes A and B appear identical. On the other hand, the patterns of fragments in lanes C and D appear unique. Are the DNA sequences and fragments in lanes A and B the same or nearly so? And is there any similarity between them and any of the fragments in lanes C and D? To answer this question, we perform what is called a Southern blot. An apparatus is set up to transfer the DNA in the gel onto a solid support, such as nitrocellulose paper. The gel is placed on top of a sponge, soaking in an alkaline solution. Nitrocellulose paper is placed on top of the gel, and a stack of paper towels is added on top of the nitrocellulose. The solution is drawn up the stack. The DNA denatures in the alkaline solution as it's carried up the stack. The DNA stops traveling when it reaches the nitrocellulose. If we could see the DNA on the nitrocellulose paper, it would show the same pattern of DNA bands as existed on the agarose gel. We place the nitrocellulose paper into a plastic bag and add a solution containing a radioactive single-stranded DNA called a probe. In this case, the probe consists of all the fragments from plasmid A. During an incubation period, the probe anneals to complementary sequences, forming base pairs with the DNA in some of the bands on the paper. The excess probe is washed off. The only probe that remains on the nitrocellulose paper is the probe that has annealed to complementary DNA on the paper. After the probe is washed off, the paper is overlaid with X-ray film. The film and nitrocellulose are kept in the dark for a period of time, and then the researcher develops the film. The radioactivity in the probe creates black bands on the X-ray film, which is called an autoradiograph. The autoradiograph provides more evidence that plasmids A and B are the same or very similar. However, plasmid D is unrelated to plasmid A, from which the radioactive probe was derived. Plasmid C has some similarity to plasmid A, but the similar sequences are found on different sized DNA fragments. The autoradiograph gives us more information than the agarose gel on its own.
So that is southern blotting. There's also western blotting, which we won't get.
These four cells represent.
All right. The second, like I said, is DNA sequences. The Sanger sequencing of DNA is the method that they use.
To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded form. A mixture containing the single-stranded DNA, DNA polymerase, the 4 deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube. When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type. The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
And there we go. I don't know why it's so long.
To sequence DNA by the chain termination.
Okay. And the last process is called DNA microarrays. These are single-stranded DNA sequences are arrayed on a solid support, or arranged can be another synonym for that. All genes in the genome are represented on a single microarray, can also be called a chip. And scientists can analyze the expression patterns of 1,000 genes in a single experiment. You said we have 20,000 genes, so this is very, very useful. To compare gene expression patterns between 2 cell types, they have to harvest mRNA from each cell type. They have to convert mRNA to a labeled cDNA, this is putting like a little tag on it, and compare hybridization patterns to sequences on a microarray for each cell type. Like I said, you'll have a different one from your brain cells than you will for your hair cells, than you will for your heart cells. And different patterns will pop up on these DNA microarrays.